Imagine anything you can do with a plastic ball. What about fighting against diseases? We are Dresden Nanormous, a team of 11 international students, and will show you what you can do with something as simple as this. Our project of a Smart Nanoreactor is based on a hollow, very stable plastic ball with just 200 nanometer diameter. The walls are made out of polymers with self-assembling properties. When their alternating hydrophilic and hydrophobic chains interact with water, they align together and independently form so-called polymersomes. So how can we create a nanoreactor out of this? DNA origami allows us to design a channel, which enables transport in and out of the polymersome. The inner diameter measures 6 nanometers, and the outside is modified with hydrophobic anchors. As a last step, our special polymers and DNA origami channels are combined with enzymes, the core of our nanoreactor. The self-assembling properties of our components enable the formation of polymersomes with the channels embedded within the walls. The enzymes are encapsulated within the polymersome and are simply too big to exit through the DNA channels. Once built, let's see the potential of our Smart Nanoreactor. Providing our polymersomes with antibodies results in a powerful drug delivery system. Once released, the nanoreactor will flow through the body, wandering in search of specific antigens and ignoring healthy tissue. However, the situation is quite different when the unhealthy cells enter the scene. Now the antibodies link to the targeted antigens, and the nanoreactor remains attached, ready for action. As a second step, an inactive form of a drug is released and flows through the whole body, homing in on the nanoreactors, without affecting healthy tissue. Once the drug reaches the unhealthy region, the polymersomes are already waiting for it. Let's follow one of them inside, through the DNA origami channel, and see what happens. Once inside the nanoreactor, the inactive prodrug is recognized by the encapsulated enzyme and converted into its active form. Now it is ready to interact with the cells and exits again through the DNA channel. Back outside, the drug starts doing its job. After a while, the task is accomplished and normalcy is restored. The pH response can control transport for molecules that are too big to fit through the DNA channel. Under basic pH, the polymersome walls are sealed, and larger particles cannot go in or out. However, in acidic pH, the porosity of the polymer walls increases, letting these particles diffuse through. This process is reversible, and the structure recovers when the pH is restored. Combining different nanoreactors within each other, we can create whole nanofactories with different compartments, which enable us to run more complex sequences of reactions. This opens a great range of possible applications in technology. Pretty smart for a plastic ball, right?